Hello and welcome to a short tour of our ION channel web resource ICGenealogy, available at icg.neurotheory.ox.ac.uk. Let's imagine that you are a neuroscientist building a biologically plausible, spatially extended neuron model for the neuron simulator. You have finished creating a passive model and now you want to add some active, voltage-dependent conductances. One option is to code a new ION channel model from scratch. Alternatively, you can use Google to find existing models. This will quickly lead you to ModelDB, a web repository that archives published neuron models, also including many models of ion channels. Here, you can search for specific ionic currents, let's say A-type potassium as an example. You will find yourself faced with an overwhelming number of around 250 possible choices. Which of these models is appropriate for the specific experiment that you are trying to simulate? To answer this, you would have to first read most of the papers for these models. And that's probably still not enough to know how these models actually behave. To be really certain, you will have to build a benchmark model to compare the dynamics of the different ion channels you're considering. This can be a lot of work. The good news is, the ICGenealogy project is doing all that work for you. We read the over 400 papers that go along with the 2,378 ion channel models available for Neuron on ModelDB and extracted relevant metadata. We then simulated all the channels in identical setups under the same voltage clamp protocols and performed analyses to find groups of channels with similar dynamics. To make all of this easily accessible, we have built a graphical browser that lets you search for specific channel models, filter them by metadata, and compare their dynamics to each other. You can even upload your own experimental data or channel models and compare them to all channels we currently have in the database. But we will get to that later. To get started, let's click on Browse up here. Everything we're going to show you is available for voltage-gated sodium, calcium, and cation channels, as well as for calcium-dependent potassium channels by clicking up here. For this video, we will focus on voltage-gated potassium, in short KV. Initially, you will find all available potassium channel models displayed as a graph. Each node here represents one model. The edges between nodes represent so-called family relations. They show how ion channel models have been derived from other previously published models. You can zoom in to look closer at specific ion channels, and you can also hover your cursor over any channel for a pop-up with basic information. If you click on a channel model, a tab will open here to the right. This contains all of the metadata we collected, and additionally shows you the current traces that we recorded from this channel. Up here you can choose to color channels by specific metadata, for example by animal model. Now you can already visually discriminate between models that were built for rodent systems and others, say cats or humans. Should you want to explore these metadata further, you can change your view, up here, switching to the XY view. Here you can choose which data to display on the axes, for example, the animal model on the x-axis and the number of citations on the y-axis. This lets you easily find, for example, the most cited channels that have been used in human or rat models. Should you then only want to consider a particular type of channel, let's again choose A-type channels, you can use the filter section down here and select Subtype and then A. You can see how easy it becomes to perform custom searches on published models, such as finding the most highly cited A-type channels for each animal model. These models here are the same 250 models that were also listed on ModelDB, but with the ICGenealogy web resource, you can now browse their metadata at your leisure. Now, let's compare some of the channels. You can select channels you are interested in by holding down the Shift key and dragging a box around them with your cursor. Then, click Compare Channels here on the lower left. This will open a new window, which provides the metadata of these channels side by side in a table. Below you can find an overlay of all the channel's current traces. Now, what if this is still not enough information to help you choose the right model for your simulations? To aid you further, we have analysed the current traces of all models by PCA and subsequent cluster analysis. This simply means that we can visualize how the dynamics of each model relates to all other models in two-dimensional plots, and also sort all channels into groups. Clicking on Similarity up here lets you view these data to easily compare the dynamics of channel models and make much more informed choices. Each of the points represents one channel model, and their positions reflect their dynamical similarity. If two channels have similar dynamics, they will be closer together. Of course, you can use the same filtering tools as before. So, let's filter again for A-type potassium channels. You will find that the same 250 channels now group neatly into around 8 groups, so-called clusters. To make this even more visible, let's colour the channels by their clusters, over here. Now, in order to find an appropriate A-type channel, you will only have to look at the dynamics of around 8 groups of very similar channel models. To make things even easier, we have selected representatives for each cluster, which you can also display by a filter down here. By shift-dragging, you can now select the 8 remaining channels and open them in the comparison tool. Finally, you can use one of the reference links to ModelDB to download the channel for use in your own simulations. A second way of displaying the grouping into clusters is the cluster view up here. Channels are now arranged on a large ring according to their similarity. Similar channels are placed closer together. The inner ring, separated by white spacings, indicates the grouping of channels into clusters. You can use this view, for example, to colour the clusters themselves by the metadata. Let's select colouring by subtype over here. You see that channels of the A subtype are mostly found here on the right. M-type channels are mostly down here, and delayed rectifiers up here on the left. As usual, you can zoom and view single channels and also filter channels using the same tools over here. Clicking on clusters gives you a detailed view of all the channels contained in them. As before, the channel comparison can also be launched from here if you shift-select channels, or even whole clusters. Finally, let's have a look at how you can upload experimental data, and also compare your own models with models in our database. Once you sign up for an account, the My ICG section becomes available. Our method relies on standardized voltage clamp protocols, so, as an experimentalist, you will need to download the time series here in Submit Your Channel. Once you have recorded the resulting current responses in your experimental setup, create a new submission and upload your files. Simply choose the appropriate protocol category in the drop-down menu here and upload each file. Uploading your own models is even easier. Simply upload your mod file and we will run a simulation of your model on our server. After upload, the submission page will get populated with graphs showing the resulting current traces. Regardless of whether your traces are of experimental origin or from a model you provided, your traces will be analysed in a matter of seconds. Once all traces are uploaded, MyICG evaluates their similarity to all channel models in the database. Down here in Results, you can find the 5 clusters most similar to your channel, together with plots comparing your model to them. For example, the plots down here show your channel's current traces compared to those of the similar clusters. You can then follow these links here to open your own channel in the comparison tool alongside all the channels contained in the similar clusters. This concludes our short tour, but we encourage you to go and explore the ICGenealogy website further. For more information, you can find an extensive manual as well as frequently asked questions in the About menu up here. Should any questions remain, you can email us using the form under Contact. Or just contact us individually. We are Bill Podlaski, Alex Seeholzer, Rajneesh Ranshan and Tim Vogels. The ICGenealogy project is a collaboration of the Centre for Neural Circuits and Behaviour at Oxford University and the Laboratory of Computational Neuroscience at the EPFL in Lausanne.